So today is the 5th lecture about the DNA sequencing, and after today that we will have 2 more lectures on the DNA sequencing side, and then will be midterm exam. Okay, haven't made that yet, but it will be ready at the time that we take the exam. So what I'm going to talk about today is pick up a little bit left over. We had 2 lectures ago before Dongbin gave a lecture on the statistical genetics. So we still have the identification for the structure variation part hasn't covered. And then I will talk about variant prioritization. Recap for the previous lectures. 2 lectures ago, we talked about variant identification. And there are different approaches that we need to do that, and which I'm not going to repeat myself. But the key point here is for different biological designs, experimental designs, your way of doing the analysis should be different. It very depends on whether it's a diploid genome balanced design, which means that you're sequencing someone's human or mouse or rat diploid genome DNA sequencing. And it is the most simplified case in the sense that you will have 2 alleles, one from father, one from mother, and then so if there is any variance, you will have the reference alternative homozygous, you will have homozygous reference variant, which is not a variant, or heterozygous, which means that 2 alleles are different, or homozygous variants, which both your father and mother alleles are different from the reference genome. So it's either 0%, 50%, or 100%. And once you have decent amount of coverage, that should be an easy case for you to figure out what the variants are. And we have discussed on that topic, and we mentioned that normally we want to have about 30x coverage to be reliable detect most of the variants. But we also discussed the ways that to increase the identification power by 2 possible ways. So the first way is to lumping multiple individuals' variant identification together, like what they have been doing for the 1,000 genomes case. In that case, it's only 4x coverage by random, but by average, but they were still able to get the variants pretty accurately. So the first way is to lumping in different individuals, multiple individuals, to do the variant identification. The second is to lumping in multiple variants within individual that they are in the LD block, so that they are forming certain type of haplotypes. And so if you don't know what I'm talking about, you better review the lecture notes. So I'm pretty sure I discussed those issues. So if this is one of the data, of course this is a fake data because the coverage is by far not enough, but I want to make the point. So if this is a diploid genome balanced design, which means someone's blood DNA or some germline samples. So you can see the reference here in this location is A, and then every single read that at this location from this particular sample is G. So you probably will be able to make the conclusion by the end of the day, this is a heterozygous variance, homozygous variance that is different from the reference allele. So if this is an unbalanced design, there's several different type of unbalanced design. So the first one is for cancer genome, which if you sequence the cancer tumor tissues sample, and in some cases your purity is not going to be, the cancer tumor purity is not going to be 100%, meaning that only a small proportion of the cells are the cancer cells. And the others are either the normal cells which hasn't become a cancer yet, or it can be stroma cells which is supporting the environment. It could be the immune cells that is within this tumor. In that case, we will call that as a normal contamination. In that case, we are not dealing with 0%, 50%, or 100% scenario. So the computational model will be very different. Depends on whether you sequence the somatic tissue with your germline control together or not. So there are different ways to do the analysis. But coming back to this, is if this is a cancer genome, and if I observed this, I probably will conclude, my conclusion will be, of course, with a much higher coverage, that this is a germline. So this is about 90% of the sequencing rates is G, which means that those are the mutations, and then so this particular one is a normal contamination. So it is possible to make those hypotheses, but without the paired control, there's no way we know that. If this is the pooled sequencing, which means that we put 10 or 20 patients' DNA together, if you observe this, you probably will conclude that most of the individuals will carry G variants at this location, but there is one individual, either one allele or two alleles, both that carries A, which is the same as the reference genome. So if this is RNA sequencing, you may think, okay, this potentially is a diploid genome, which at this location is a heterozygous variants, A or G, but G allele, it will express much higher level comparing to the A allele. So this potentially can be the unbalanced design. And we also talk about variant identification based on sequences from multiple individuals. Okay, that is what we talked about two lectures ago. And there's another point in addition to the variant identification is there is a variant quality recalibration. The reason that we need to do this is after we identify variants, for every single variant location, there are many different parameters that we can tune on. For example, if I want to making sure that all my variance is larger than 20x coverage, so that it will be a threshold. Or I want microbility is strand bias is not significant. So there are many different descriptions on this particular variant locations. If you really want to filter out, you want to leave throw away the bad variants, the missed cause, and in that case, you probably will put a lot of threshold there, and you say, okay, I want the variant, the sequencing depth is larger than 20, so you have one parameter. You want the mapping quality average is larger than 20. There's another threshold. So you can set up a dozen of those thresholds. So the problem for that is it become very arbitrary, and then sometimes the reproducibility across different groups will be very different. So for this variant recalibration, what they have been doing is they were trying to calculate one score that is describing this overall variant quality of this particular variant. And then only set up one threshold to do the cutoff. So the way they do that is they do the training on high confidence variants. So basically, if you identify for the whole exome sequencing, let's say you identified 40,000 variants. And then by the end of the day, if you're overlapping these variants with DB-SNP, you found 35,000 of those already been found in the DB-SNP, which means that you're very confident on these variants. So now let's learn from these variants, and then to look at overall this data distribution about those a few dozen features. And then you train a model, and then you develop a machine learning tool. So for every single variant that is not being seen in the DB-SNP, and then you run through this model, and then it will tell you that what's the likelihood this variant is a true one or not. So that is the variant recalibration side, and these are the part we discussed about 2 weeks ago. Okay, so these slides, although very simple, but summarize the major components that we need to think through when we do the DNA sequencing analysis. So this will start from the raw data, and then we will need to do the quality assessment and to see whether the variance is good or bad. And there is a part that in the a couple of lectures ago, we talked about the base quality recalibration. And then we need to do the sequence alignment, align those reads back to the reference genome. And then we need to do the refined alignment. Remember those variants that are very close to the small insertion deletion sites. And then we do the variant calling like we discussed last time. And after that, it will be the variant calling recalibration. So those are the ones that we have discussed. And then today, we will touch base on the functional prediction, which means which variant you want to focus on for one of the disease settings. And we'll go through the bioinformatics prioritization on this part. And there is also the rare allele association analysis. And Dr. Lai discussed about some of this topic about the burden test and a few other approaches in the last lecture. I do expect you to getting familiar with all those materials being covered in the last lecture as well. Okay, so let's now shift our gear to the structure variation identification. Okay, so first. And these are different type of large structure variations. So when we talk about the structure variation, normally people are talking about the large ones, larger than a few thousand bases, and sometimes even bigger than that. So here is a summary of what are the most common large structure variations that are out there. So the first one is deletion. So you can see in the reference genome, and this is your particular donor genome. And very clearly that this part has been deleted out in this particular donor. So you can see that these two sets, they do not supposed to come together, but somehow they do come together. So in the next slides, I will tell you that what are the information we can use about this information. And then the second mode will be novel sequencing insertion. So you can see this is the reference genome, but in this part, there is an insertion in this. And this insertion is actually pretty novel. We don't know where they come from. So actually in the initial, very early phase of the reference genome, they do have some deletion. Those reference genomes have some deletion. So later people found out there's many large insertions is part of the genome, and then eventually concluded in the reference genome that those were the deletions. And those are the parts that were later basically fixed in the later versions of human genomes. So this is the third mode is pretty similar to the second one. It's also large insertions, but the difference is that this middle part will be the mobile elements and those jumping around in the genome. So this type is called tandem duplication. So you can see that this is one part of a larger stretch of DNA sequence. They were being duplicated multiple times in the genome, in the specific locations. So they are kind of connecting together. And if this duplication occurs in different locations and with inserting different part of the genome, this is called interspersed duplications. And you like it or not, all these variants really happens in our genome, both in the germlines and in the somatic tissues. And this is the type is called inversion. So you can see that this part is the same as the reference genome. This part is the same as the reference genome, but somehow the middle part, it turned the direction from this side to this side. And there is also translocation, so that there's one part of the elements is supposed to be in one part of the genome, but somehow it's moved to a different type of part of the genome. So, I mean, I'm pretty sure this is an incomplete list of all the large structure variations. So there are kind of more complicated ones. And before we have the next generation sequencing technology, it almost become impossible to identify these larger structure variations in a de novo fashion. So you can, for some known ones, and we already know that they are in certain part of the genome, like 1Q in the multiple myeloma, or 17Q in some of the part, you can use FISH technology to pull down those regions and to try to identify those. But if you want to find the large structure variation in a de novo fashion, it's almost impossible. With the next generation sequencing technology, and this start to become improved, although we cannot hope that we can completely exhaust our search for every single piece of it, because it's a very much a customized way of identifying those. But in general, so I did put in this paper that I don't remember whether I got this from this paper, probably not. But you can see that these are the different type of information that people are using to identify the larger structure variations. In general, there are 4 different type of approaches. There are probably a few dozen of those algorithms trying to identify these large structure variations, but in general, they are focusing on these 4 type of information. The first one is the read pair, and we know that when we do the DNA sequencing, the normal practice is to use paired end arrays to do the sequencing. Normally, it's 150 PE, so you got a DNA element that is fragmented into about 500 bases in a piece, and they'll sequence one end 150 base, the other one 150 base. So that is one level of information that we will use. That is called a read pair. And the second one is the read depth. So just counting. So for example, in this case, I know it's small, but we'll have a bigger version in the next slides. But you can look at here is if there is a deletion in one of the alleles, that you are expecting to see a much lower coverage in those regions. So those are based on the read depths. For this one, if you are seeing some of the duplication, so you can see that and either interspersed or tandem duplication, so you can see this part, if you align back to reference genome, you see much higher coverage here. So that is the read depth. The third part is called split read. So basically, we are still relying on the paired end sequencing. However, for every single read, that this is most likely those reads will cover a junction of the structure variation part. So we'll get into a little bit more. And the last one is assembly based, and to basically do the de novo assembly on these regions. So let's take a look at break this down one by one. Again, I'm not in the process of telling you any specific algorithm what to do with it. Just broadly in general, what type of information people are using to do the large structure variation identification. So the first type of signal is the read pairs. So you can see this one is the deletion, and this is the insertion, and this is the inversion. In this case, what we normally do is we'll first assess the distance and the orientation with the paired end arrays. For example, if our initial assay is this DNA fragment is by average about 500 bases give and take. And then if we have paired end sequencing, meaning sequence 100 or 150 base pair into each direction, okay, if we map this back to the genome, normally we will see that the distance between these two will be about 500 bases, right? It cannot go too far away or too small. I mean, you can have a certain type of variability, but it cannot be like 3,000 bases away, because that's not how the experiment was done. But in this case, if you map these reads back to the genome, and one end mapped to here, the other end mapped to here, and then you found out these two ends, their distance is actually 5,000 bases. And that is not happening because it's not possible, because your assay, your DNA fragment is only by average 500. So the only conclusion for this, or possible fashion that this will occur, is there is a big deletion in the middle. Okay, so that is the type of information people will use. In this case, this will be the insertion size, but this is more limited because in many cases, this will be much, much smaller. Sometimes they even run into each other. And then for the inversion side, so you can see that this is pretty much not only based on the distance between these two ends, but also the orientations of the two ends. So you can spend some time to read through this and figure out what this really means. But in general, by evaluating the distance between these two ends, and as well as the orientation between these two ends, and that is the amount of information people are using to identify the structure variations. So basically, you assess the distance and the orientation of the pair and the rays, and you cluster the discordant pairs in which mapping distance or orientation of rate pairs are inconsistent with reference genome. So basically, those are called the inconsistent discordant pairs. And then you do some level of statistical estimation to evaluate the distance between these two rays, and to see what's the likelihood there is a large structure variation, deletion or insertion in the middle. Any question? Does that make sense? Okay. All right. I see some of you nodded, so I appreciate that. In general, this will be capable of detecting multiple types of structure variations. And the limitation of this is it cannot capture small variants, because if your deletion size, for example, is 5,000 bases, it's pretty obvious. But if your deletion size is like a 50 base, you really don't know whether that added the distance on the reference genome is due to there is a deletion in the middle, or it's because you happen to grab a bigger DNA fragment to do the sequencing. So that is not doing very well for the small variants. And for the very large insertions, sometimes it will become difficult as well, as you can see that in this case, that normally the two ends is basically about 500 bases apart, and it's not possible you have a very big insertion in the middle, so you can still be able to detect using this way. Okay. The second type of information people are using is the split read. Okay, so this is not relying on the paired end, both two ends and their distance and orientation. Rather, and this is using one of the two ends. So you can see that when the structure variation junction occurred within these fragments, and then if you do the analysis properly, you will be able to identify those structure variations, right? For deletion, in this case, that you can see that this happens to be that one sequencing read, one side, so not a pair, okay? And in the previous one was a pair. So now one of the two, and then happens to be, when they map back to the genome, so you can see this read is being broken into two, and one half is mapped in this location, and the other half in this location. And this will be a very strong indication that in the middle part there is a big deletion. Deletion is always a little easier to identify compared to other type of events. And in terms of novel sequence insertion, and then you can see this read, sometimes if it's not very big, And then you can see that this read one part mapped to here, the other part mapped to here, but somehow in the middle there is a third element that you have not seen before, and those are more likely to be insertions. But this type of variance, it cannot be very big insertion. It has to be very, very small, and small enough to be integrated into one specific nucleotide read. And in this case, that will be annotated transposons. So you can see that this transposon element is actually jumping into here. And then if you map it, it's one end is here, the other one is here, being mapped to here, but actually it's the transposon elements. If you only rely on this one particular read, you may calculated this is as a deletion. So, but you need to pair those with the other region in terms of the overall reads. But you can see this is the type of events that could happen as well. So I'm not going to go through the remaining ones, but the point is for the split read is your structure variation junction occurred in the middle of one of the two reads. So if you analyze the data properly, this will help you to identify different type of structure variation. The next part of this split read analysis is, first of all, it's harder. So you need to write your program in a specific way to be able to identify this. But second, the advantage of this is if you were able to identify, actually not only to estimate there is a few hundred base insertion in the middle or deletion in the middle, but you know where it happened. You know exactly what the junction is, because your one end here is mapped to here, the other end mapped to here. So you know that this is exactly the site of the deletion occurred. Well, if you were based on the previous one, the read pairs, you can only estimate that there's something happened in the middle by looking at the distance and orientations of these two reads. As I mentioned, the third type of information people are using is the read depths. So you can see that for deletion, for the interspersed duplication, tandem duplications, and in this case, you will be able to see that there is a lower or higher coverage at a certain part of the genome. So you will be able to identify there is a insert deletion or duplication occurred in those regions. For this one to do this, you need to be a little bit careful. And we didn't talk about too much on the experimental side. When you do the DNA sequencing, there are multiple ways of doing that. There is a PCR-free ways. There are the ways that you can use the PCR. And if your goal is to identify this, the recommendation will be you use the PCR-free way to do it. So meaning that otherwise, some of this may coming from just the PCR duplication is more uneven compared to other regions. So that can cause some issues. And there are specific tools being written to identify this type of structure variation based on read depths. For whole exome sequencing, there are a bunch of tools are written here. And then for whole genome sequencing, there are different tools. Okay, I'm not going to go detail of any of individual ones. There are ways that, so this is the paper that talking about one very popular bioinformatics tool to do the structure variation called the LUMPy. And what happens is it basically integrated all this information together and using the information we discussed in the earlier slides. So this is a kind of a pretty gold standard of doing the structure variation identification. Of course, there are continue to have newer ones that coming out for this type of business. Okay, any question?
So what does breakpoints just mean overlapping reads?
The breakpoints. Which one? This one? This. This one?
In the last, like, on this slide,
Oh, in here?
there's a breakpoint.
Where is the breakpoint? Oh, here. Is that breakpoint evidence?
Yeah.
What's your question?
Is it just the overlapping reads?
No, it's basically it try to collect all the evidence for the breakpoint and for the read pairs, it won't allow you to directly identify the individual breakpoint directly, but it will provide a certain level of confidence on certain breakpoints that could be supported by the split rays, right? So you always have some of the rays are split rays, others are basically the paired end rays you can look into. Yes?
Can we go back to the previous one? So when we look at the deletions, there might be some leads which might be split between
Yes.
there's a region which is deleted, so some part of the lead is on this side and on the other side.
Yeah, absolutely. This is one type of information people can use. So that doesn't mean this is the only information you use. Meaning that if you do the deletion identification, most likely that some of the reads information you are going to use is the read depths like this or this. And there will be some of the paired ones that you can see some of the evidence based on their distance after doing the sequence alignment. And there will be the one that on the split reads that will directly one end goes to here, one end goes to here. So all the information will be used, but this is one type of information people are using. One other point is, so this figure is plotted in the way everything is continuous on the genome, right? And sometimes this will fit for the whole genome sequencing type of analysis. But if this is a whole exome sequencing, and different exons may not connecting together, so the breakpoint may not be feasible to do. So in that case, the read depth is the primary information you'll be using for this. Other questions? Okay. So for the structure variation, there is another level of signal that or approach that we can use to tackle this a little better. And so this is the third straight lecture I brought up, this 10x linked read sequencing. Just refresh your memory a little bit. And so this is the way that you can see this is one specific gel. And then in this gel, you will track about 10 molecules. Each one is about 50,000 bases. So it's very, very long. It's not 500 at all. But what you do here is for all these DNA sequences, you are going to have a barcode specific, in this case, the green barcode that corresponding to this particular gel. Okay. So, and also that it's unlikely that we will sequence the entire 50,000 base sequence. Rather, for each individual fragment, it's about 20% of the region will be covered by this. So that is what the technology is. This is the technology. If we use this to do the DNA sequencing, actually, we mentioned it will increase the mapability dramatically. And then we also mentioned that it will help us to identify the haplotypes, meaning that lumping multiple variant sites together and knowing which allele they're coming from. But their actually major advantage is to do the structure variation identification. So I'm just showing you one example here. And then you can see this is the genomic DNA region 1 and 2. So this is your reference DNA, and there is a 50,000 base, which is very big potential structure variation that is in the middle. Okay, so if in the case of there's no structure variation, okay, and in that case we will see that some of this, keep in mind each fragment here is about 50,000 base, or 50,000 to 100,000 bases. So you can see that we see a lot of read coverage that when they are close together, they will share the same barcode, same color barcode. And this will be the same color barcode, this will be the same color barcode, right? And if we pick up these two regions, it's unlikely they will share barcode because they are very far away. And this whole middle part is like a 50,000 bases. It's already pretty far. But if there is a deletion here, so what you have is these two parts, there are 50,000 base in this donor genome is supposed to be connecting together. In that case, when you do the long-range DNA sequencing, you will see the sequencing rate will align in this way. But once you map it back to the reference genome, we know that they are being breakers. So one end is mapped to here, the other one is mapped here, and then in the middle there is no sequencing reads at all because for this individual there is a deletion. So in that case, we will see a very high level of barcode sharing across these two things. And these two regions, unexpected high level of barcode sharing. If that is the indication there is a deletion happened. Okay, keep in mind, I'm showing you here is only for one allele. Okay, and when we do the sequencing, and we actually sequence both two alleles. So in that case, it could be that some of the reads that we see this level of sharing very high, but some of the reads they are not sharing at all because one of the alleles having this 50,000 base in that. Okay, so in this case, the data that we are observing here is if there's no structure variation, these two parts doesn't have any barcode sharing, but once there is a deletion, there will be high level of barcode sharing. So those summarize the previous slide. So let me show you one of the results that when they told us. So this is something that you use a regular approach to do the DNA sequencing, and then you will see that here is your read map, right? So you can see there is some up and down, meaning those are the coverage, and those colorful ones are normally there is a SNP that occurred in those regions. But if you look at this, there is about 1,200 bases deletion in this region. But from the coverage, you really cannot very reliable say there is a deletion here, right? So basically you're stuck there. You can have in trouble whether this is a deletion or not. But if we did the long range linked read sequencing, so what we can do is we can recover each individual read to know which allele it comes from. So in that case, once we map it back to the genome and then color code it based on the facing information, which allele it belongs to, so you can see this is the part that is only the blue allele show the coverage, none coming from the red allele. And then what you can do is in the IGV, you can base on this allele tag to break those into two different tracks. As you can see, in one allele, there is clearly a deletion. In the other allele, there's no deletion. So those are the information that we can use to help us identify structure variations. Actually, it gives much higher sensitivity for the structure variation identification. And the last slide in the structure variation is there for the short read sequencing, there is always an issue, regardless of what tools that we are using. So whether we use the split reads or we use the read pairs, we use the coverage, we use the assembly approach, the power is very limited. It's just because of the short read nature of this. It's not adapted to identify structure variations. A true fix of this issue will be using the long read sequencing, and that could be somehow that the linked reads type of technology that can help us with that. But it will be much, much better if the sequencing technology itself can go longer. So, for example, if we use Pacific Biosciences or Oxford Nanopore sequencing, and that will help us to directly sequence a much higher extended region so that it will increase our ability to identify structural variations. And there is a trend that people, actually I talked with PacBio people a couple of weeks ago, and but from what I hear from them is at least currently to sequence using PacBio is still very, very expensive. And especially if you want to sequence more accurately. If you don't want to sequence very accurately, it's fine to be a little cheaper. So there is actually a trend that you use a combined approach. You sequence the same DNA with PacBio technology and doing a little bit shadowed sequencing, and then you use the second generation, the Illumina technology, to further correct these rays. And that actually works well for some of the approach to identify structural variations. Okay? Any questions so far about structural variation? No? Okay. So now I'm going to switch the gear to variant prioritization. So what we're trying to figure out here, okay? You have some DNA, and then you grab it, you extract, get a sample, you extract the DNA, you did the sequencing, and then you go through all the steps that we went through, okay? You did everything right, like we did quality control, quality recalibration, and then sequence alignment, refined alignment, variant identification, variant recalibration, and then statistical analysis. You did everything. By the end of the day, you identify 10,000 variants. Okay, so see, this is potentially associated to the disease, and I'm very confident that these 10,000 variants are real. Okay, then what you do about it? And we know that for most of these variants from the very early lectures, most of them are so-called passenger variants or mutations, which means they are just sitting there. They are not doing any harm or influencing your phenotype in any way, but they're just sitting there so that you cannot find out the driver mutations. So in order to do that, we want to see what the real question we want to answer is which variant can potentially cause the phenotype to be different. And that is something that we want to bring back and then to understand what these variants are doing. So then one step that we need to go through is going to be a bioinformatics prediction, which is a variant prioritization. So in this lecture, this part, I'm going to review what are the tools that people are using here and what are the information they have been using to do this. Okay. So when you do the variant prioritization, there are several different levels. The first one is you basically just do a variant annotation. So what that means is I need to know where they are, right? Which variant, is that in the coding region? Is that in the intron region? Is that in the exons? Is that in junction? Is that in the 3' UTR regions so that they can potentially be influencing the microRNA targeting, or is that in the 5' UTR that they can potentially influence the translational control? Is that in the non-coding RNA? Is that in the promoter regions, the enhancer regions? So regardless of what you do with it, you have to do a level of annotation to see which part of a genomic region they are. And the second level of information people normally use is to look into the population-based annotation. So which means whether one specific variant, I have seen those before, okay? So let's say if you are working on a disease that is early onset disease, and it's devastating, but it's early onset. And the chances, if a variant that is causing this disease is unlikely to be seen in the general population. Right? So if everybody carry it, And if this is a very rare disease and early onset, the chances you wouldn't be able to identify that in the general population. So that's why that one important step people do is we'll look at whether we have seen that in the DB-SNP, whether we have seen that in the 1,000 Genomes Project, and because those are healthy individuals, if the specific phenotype that you're looking at and it's not it shouldn't happen a lot in the general population. So which means that sometimes we want it to be like less than 5% or sometimes even 1% of allele frequency in the general population, and whether it's in other databases as well. So those are the level of information that we want to use. But we have to be careful on this approach. Is this really depends on what type of disease you are working on. As I mentioned earlier, if you are working on early onset childhood cancer, and the chance is you wouldn't find the driver mutations in the general population. But if you are trying to look into like a hypertension or Alzheimer's disease or Parkinson, okay, those late onset disease, there is a good chance that those initial 1,000 genome samples, they are healthy individuals, but they have not been old enough to see those phenotypes yet. So it really depends on the disease that you're working on and how much aggressive you are in terms of filtering on this. And then the next level of information we want to look at is based on their molecular functions and on those individual variants. First of all, the most important part people always look at is whether it will change the protein structures. And if it changes the protein structures, you better change the amino acid sequences, which we'll go through those theories a little bit. And the second part is to look for whether those variants can potentially cause splicing changes. For example, if some of the variants are locating on the splicing site, or whether it's within an alternative splice exon, and whether some of the synonymous variations, even though they are not changing the amino acid sequences, they could change the way that splicing happens. So we'll give you some of the examples of that. And the last part will be whether these variants can play any role in the general gene regulation. For example, if a variant occurred in the promoter region, it can impact the transcription factor binding sites, or if it's in the 3' UTR regions, it can impact the microRNA binding sites. So those are the information we'll look at to help us to prioritize which variants are potentially useful. Okay, so the first part, which is the annotation based on genomic loci. So here are the listed loci people often look at, whether it's in the exon, intron, 5 or 3' UTRs, microRNAs, either in the precursor or mature, and so on and so forth. So there could be multiple different annotations you want to look at. And the major sources for this is you can look at the RefSeq database, UCSC gene database, Ensemble database, and GeneCode. There are multiple annotation databases available. So the second level of genomic loci-based annotation will be looking at the evolution conservation. And that has become a very popular tool that people are using. If you identify variants, you want to see whether this variant this loci is conserved across multiple vertebrate animals at the very least. And the underlying assumption of that is conserved regions are more likely to be functionally important. So if it's conserved in many different species, they are more likely to be important. But in this, and sometimes we also need to be careful because there are some of the disease or some of the functions that is more specific to humans. And those are called fast evolved regions. So meaning that only in the primate or in the human that has those regions start to show up functions. And for those of the regions, they can be very, very important, but only if we do the filtering based on the evolution conservation, they actually might be missed. And we also need to rely on, in terms of functional annotation, and rely on other type of functional genomics data. For example, whether transcripts that are expressed in the organ or tissue that are most relevant to disease. For example, if you want to study breast cancer, for example, and then you better this gene needs to be expressed in breast. And if it doesn't, the chances it may not be super useful. This sometimes, however, can be difficult because what if that gene does not express in normal breast, but rather express heavily in the breast cancer? That could be the driver genes. So sometimes it can be misleading. And also, in many cases, we actually don't know which organ or tissue to look into, right? And for example, and some of them, for example, hypertension, right? Which tissue you look at? There could be many different tissues that are being pathogenic. And some of the disease you think you know, like Alzheimer's disease, right? And in the past, people looking at the neuron or some of the tissues in the brains, but more and more evidence suggests the immune system has a lot to do with Alzheimer's disease. So there's a lot of things that could be misleading. Well, so we just need to be careful. This will lay on an additional layer of information that help us, but we cannot purely rely on those. And that's what I was saying always, that you need to know that what are the assumptions of all different approaches. And another level of information will be the promoter and enhancer and the DNA hypersensitivity regions. And those are the functional genomics regions that has been available through various projects like ENCODE or epigenome roadmap projects. And those can be very, very useful to help us to see, let's focus on the SNPs in the promoter regions, for example. And most of this information, it sounds a lot, but most of these informations are actually available in the UCSC genome browser and in many different databases. Okay, so the annotation based on the population, I mentioned earlier that dbSNP will be a very important source. So I will just briefly talk a few words about dbSNP. It's hosted by NCBI. It actually called dbSNP, but include more than SNPs. It has indels, microsatellites, and many other type of genetic variations in that. And the records include more than human, actually. It has 55 different organisms. And all the data from the 1,000 genomes and many data from many sequencing, big sequencing projects is already included in the dbSNP. And these are the 1,000 genomes one, and there's another one which is exactly the database. So those are the information that people often use to annotate what are the allele frequency in different populations. And if, again, the rationale behind that is, and if one variant is causing certain disease, and that variance doesn't supposed to occur in a very high level allele frequency in the general population. And I have repeatedly saying that we need to be very careful. It depends on the disease to be used. Okay. Another very useful database is called CleanVar, and this is another effort that by the NCBI. So you can see that the archive the variants that they know to have a clinical impact to the phenotypes, and those variants are typically being reported by the clinical laboratories, and then they were being put into these databases. So I'm not going to go into detail, but you can go there to take a look at. There's a searching functionalities, And sometimes they also have different levels of confidence, because every time that when this opens to the public, the chance is there will be a lot of noise in that, right? Sometimes that we know, as always in the scientific community, we may have conflicting results. And so actually in the claim bar, for every variation, they will have different levels. Which one has been validated, curated by the NCBI staff? So those are normally the highest level of confidence for those variants. Some of them, although they are not being specific, independently validated, but they have been reported by more than one clinical laboratories. So those are the ones that potentially will be more confidence. And then, but some of them have conflicting results. So you will see all those informations in the report. You can choose to believe one way or the other. So those are the annotation based on the population or based on existing databases. And there is also a very strong effort to look at these variants based on the molecular functions. And some of them are nonsense variants, meaning that they're within an exon and translated amino acid to change the stop codon. So by the end of the day, if you have such variant happen, you will have a truncated protein, or you will have a much lower expressed gene, because that could trigger the nonsense mediated decay pathway to kill the RNA molecule. Sometimes there are variants that could be frame shift, and those can be annotated as well. And if this coding variants, whether this variant is non-synonymous SNP, meaning whether this variant is nucleotide changes will cause the amino acid sequence changes. So those are important parameters that we look at. Whether it will impact directly on the splicing junction sites, because those are the regions that can possibly impact the splicing outcomes. And if it's in the coding region, in addition to non-synonymous SNPs, there are some of the SNPs that they could be synonymous, which means that they are not changing amino acid sequences. A lot of people will consider these variants as a silenced variants, which means they are not causing any harm because they are not changing amino acid sequence. But I will show you some of our own study by showing that some of these are very important in the splicing regulation as well. The variants on the 5' or 3' UTR regions and those near gene regions, which can be on the enhancer regions or promoters, and then some of them are in the intronic region. So all those are different levels of annotation based on their molecular functions. Okay, so now let's discuss a little bit more on the bioinformatics prediction side. Okay, so it's very possible that many of these variants are called non-synonymous variants, which means those are the nucleotide change will cause amino acid sequence changes. They are normally considered as very important. They constitute more than 50% of mutations known to be involved in human disease, and based on various databases. And in human populations, there are about anywhere from about 200,000 common variants. They are non-synonymous SNPs, so meaning they will change amino acid sequence. And I think I mentioned earlier that in James Watson's genome that they had done back in 2007, they were able to identify 11,000 non-synonymous SNPs in one genome with a very low coverage. So in general, that per person, we are expecting somewhere about 40,000 heterozygous non-synonymous SNPs in humans. And there's another important factor is non-synonymous SNPs tends to have a lower minor allele frequency distribution than those are predicted to be functionally neutral. So what that means is in general population, non-synonymous SNPs occurred less frequent compared to other type of SNPs because they are functionally important. So evolutionarily, we cannot afford to to change them. So once some of those are being altered, and that means the protein function are changed, so the organism may not be fit to survive. So that's why that functional variants tends to have a lower allele frequency in the general population. That is the rationale for that. And so what are the general strategies for predicting the functions of non-synonymous SNPs? So some people will say, okay, so look at the non-synonymous SNPs. I mean, if you change the amino acid sequence, it got to be important. Well, not necessarily true. So some of the amino acid sequence change will not cause a huge impact on the protein functions, so they may not be very important. So therefore, there is a strong need. So we know that for humans, for every single one, we're expecting about 40,000 non-synonymous SNPs. And it's unlikely that every single one of them will be useful. In that case, we need to do the functional prioritization for the non-synonymous SNPs. And so you can see that in this conclusion, disease-causing mutations are more likely to occur at the positions that are conserved throughout the evolution as compared with the positions that are not conserved. So here is some of the figures. So you can see the top one here is there are 3 different lines. Okay, I want you to pay attention to the red line. Red lines are the disease-causing mutations. So those are functional ones. X here is the conservation. So you can see that disease-causing ones tends to have a much higher conservation and across multiple species. Okay, so this is in line with what I told you in the earlier slides. So there is a conservation, evolution conservation that we need to look at. And for the bottom one here is, this is called the solvent accessibility of mutation sites. So this means that whether this site, so we know that proteins, they can form secondary, tertiary structures, right? So they form structures. And people did a calculation. They found out that disease-causing mutations actually tends to locate inside of the protein rather than on the surface side of the protein. Okay, I think this makes sense because if it's at the surface, and in many cases it will just impact the local structure. Well, if it's inside, and especially when the charge or the size altered dramatically, and that will causing the entire protein structure to be disformed or form in a different way. So that is one of the reasons that why the SNPs are inside of the protein structures can be more important. So given this said, that put this into a perspective, so there are several different type of information people are using to prioritize the non-synonymous SNPs. So there are mostly 2 types. One is the sequence information, and including the evolution conservation and the sequence alignment, as well as the structure, the protein structure information. Of course, if there is a more known about the annotation of those individual regions, that will be useful as well. So there are many tools that has been developed, and this is a kind of a very much settled area. So you can see that there's a very thorough review back in 2006. So many years ago, people have been working on this, and there's not too much improvement afterwards. So there's not too much. I mean, it's considered one of the areas that is solved. Well, it may not be fully solved, but people cannot do better. So when you do the using different algorithms, the most famous algorithm that people are using is SIFT and polyphen to predict the functions of non-synonymous SNPs. Have any of you heard about that? SIFT and polyphen? Okay. Just go to look at it. Those are the part that you cannot escape when you do the variant prioritization for the non-synonymous SNPs. So, what I want to mention briefly here is SIFT is primarily using the sequencing information, while polyphen use both the sequence information and the structure information. Okay, so those are the something that we need to pay attention to. The next question is, there are so many algorithms on that, and which one should we use? And actually, direct comparison among different methods are very difficult because they are all based on very different assumptions. And the performance is depending on your training data and the testing site. And if you publish a paper, there's always a way that you can show that your method works better than the others. So it's not necessarily to be completely fair. But we need to understand that the caveats of different methods. So you will say that if we do the structure-based analysis, not based on the sequence-based, and you will think, oh, that will be more useful because the structure is based on the sequence. It actually has more elements that being chipped in. I mean, that can help you to increase your statistical power or prediction accuracy. But we need to making sure that we appreciate the fact that if you do the structure-based prediction, and normally you will have less prediction available because not every protein has a structure available. Some of the proteins doesn't even form the structure. So in that case, structure-based analysis will not be available for everything. And then also that sometimes it can be misleading because the structures are predicted as silos. Normally it's not what the real structure is in the physiological condition. It's what happens in the tube, and so it's unclear whether they fully represent what happens inside the cell. So that is the caveats of the structure-based study. Well, for sequence-based study, that they were based on the homology and the orthologs of the same sequence elements in different species. But one thing that we need to know that there is no clear winner about which algorithm to use. So in general, what people do is we will try all of them or a few of them and to see what are the ones that are being selected by most of this. Again, this is not the best practice that possible, but that is something we have to do. And another point is these predictions are good for guiding the experimental design and to say, okay, if there's a variance that you think is very important, and then you cannot use that as a clinical decision supporting mechanism. Okay. And it's simply just not allowed. It's not advised. So what that give you is simply a hypothesis that you can test. You can make in animals or making the cell line models and do the CRISPR to change that mutation to see whether the phenotype of the cell or the animals that change the behavior or not. So those are, again, a research tool. It cannot be used in the clinical settings. There are a few tools that recently published focus on combining different tools together. So we have the SIFT, we have polyphen, we have so many different algorithms that people can use, and none of them are clear winner of one way or the other. So what happens is people will try all of them and then using a machine learning based approach to give an overall ensemble based approach. So this CADD or combined annotation dependent depletion analysis will be one of the tools for that. So once you feed in a SNP, and it will give you a CAD score, CADD score, and that will help you to, summarize many different tools and then give you one final score to do the prioritization. Okay, so most things that I talk about so far is based on the non-synonymous SNPs, which means that those are the SNPs that are changing the amino acid sequence. But you probably know that after ENCODE project or after GWAS, genome-wide association studies, and people found out actually over 90% of the disease-associated variants are located in non-coding regions. So they have nothing to do with the protein coding sequences. And how come that they can have a dramatic impact on the complex disease? And the reason for that is most of those variants, functional variants, are actually something called a regulatory variants. So meaning that they are not necessarily changing the amino acid sequence or change the protein function. Rather, they will change the regulation of these genes by altering their gene expression levels, by altering their RNA degradation, by altering their splicing patterns. And so I will briefly go through the examples of this type of variants so we can see that how that will help us to try to search for the functional genetic variants. Yes?
These variants are not considered in non-synonymous, like they're not considered non-synonymous variants?
Which part?
The one which is predicted.
Which I mentioned for the regulatory variants. No, they are not considered. So non-synonymous variants have very specific definition. Those are the nucleotide variants that will change the amino acid sequence. Okay, so those are the ones that most people are looking at. But more and more people start to looking into from a different angle to see the regulatory variants. The variants that will cause the regulation change of the gene expression levels. And this will connect well with the lecture on next Monday by Dr. Xi Rao and talking about functional validation of larger scale functional screening of those variants, their potential functions. So I will talk about first on the informatics part, and she will connect next week on the experimental side. Yes, there's a question.
So I can see how synonymous SNPs will affect expression and function, but I cannot understand how it will have an impact on the conformation of the protein.
On the conformation of the proteins? Yeah, so we'll get into that. Okay. So those are synonymous SNPs. So let's focus on these regulatory variants one by one. Synonymous SNPs are obviously still in the coding regions, and it's not in that 90% of the non-coding regions. But this is another part of the variants that people often ignore. They are considered as silent mutations because they may cause, they are not causing amino acid sequence changes, but they may change the protein expression, conformation, and function. So we get into that, what are the potential functions. So they were considered has no effect on fitness of organism or would be neutral during the evolution. And actually this turned out to be not true, okay? Actually a lot of the synonymous SNPs can be very, very important. So I'm just showing you some of the previous studies on this. So this is from this paper. This is kind of early, and when GWAS, the first round of a couple of rounds of GWAS study come out, and for this study, this is from Attribute group from Stanford, and they basically look at over 2,100 different studies, and on 21,000 documented associations between SNP and disease. Here's the conclusion. So non-synonymous and synonymous coding SNPs show similar likelihood and effect size of human disease association. So here is some of the detailed data. So you can see that I'm pointing out that the left ones are the likelihood of disease association, and the Y here is the effect size. Okay, so if you will, this is p-value, this is the effect size. But what I want you to point out here is this column here is the non-synonymous SNP. the SNP that we think could change amino acid sequencing must be important. And this is the synonymous SNP. So you can see they are pretty same level of bars. The likelihood of disease association is almost identical, and then their effect size, there's no significant differences as well. Okay, so this is another study that coming in Cell that surveyed a lot of cancer genomes. So here's the conclusion. Synonymous mutations frequently act as driver mutations in human cancers. And I'm not going to go into detail. They have made some conclusion about the oncogene and tumor suppressors. But the point of all these studies is they are synonymous SNPs is important. Why is that? Okay, so this is coming back to the potential mechanism. So you can see that this is the most possible simplified model of this. I'm sure this is not how it works, but this is a very simplified case. So in this case, that there are 3 proteins. There are SR proteins. They bind on exon regions. And then once they bind here, the splicing will occur here. So you can see that end up with 3 different protein products and exons. And then for this one, if there's a mutation occurred, and this protein cannot bind, in some of the cases, the splicing only occurs here. So you can see if that's the case, the middle part will be spliced away. So even for this one single individual SNP, and it doesn't change amino acid sequence, but it could cause the entire protein domain to be missing. Okay, so that can potentially lead into a larger physiological impact. And another possible way that this work will through the control for the global stability and the local stability. So you can see this is the figure that we know that RNA, they form secondary structures. Okay, so these two are almost identical in terms of the structure, except for this part. There is a SNP. And if there's a SNP, and this part will not be perfectly paired together. And then in this case, this will generate something like this. And this is normally a less stable form of the RNA molecule, so that can be degraded more than the other one. So this is one of the mechanisms that this synonymous SNP can potentially change that as well. And so this is the part that, so you can see this part is actually near the start codon. This is SNP occurred in near the start codon. In this case, you see this double stranded part will be tightened up together, more sticky than this side. So which means the translation may start easier from this type of molecule compared to this one. So meaning that you may end up with a more protein product. But the point of this is there are many different ways that those SNPs is going to impact the splicing regulation, the translational initiation, RNA stability. So this is another one that is on the codon usage. And there's a couple of years ago that there is a science paper talking about this. So basically, it could be these two, they are encoded for, the exactly same amino acid. And so this is 3 nucleotide will encode for the same amino acid. But however, if we have more of the red ones compared to the yellow ones, and those red tRNA complex will be more readily available, so that the translation efficiency will be different. So although that this variance is not changing amino acid sequence, but it will change the translational rate by controlling the translation efficiency. So again, that all these different ways that possibility that this individual SNP is going to change the gene regulation in many different levels. Okay, so post-transcriptional regulation, translational control, and proteostasis, and so on. Okay, So now we can, well, I'll tell you that, for some of the methods that to look for the predictions of these synonymous SNPs, how do we predict? So if we have like 30,000 synonymous SNPs, how do we predict which one is likely to be more important? And here are the information people often look at. It's distance from the splicing site. So it's closer to the splicing site that has a more impact on the splicing regulation. Whether it will affect the forming of the spliceosome, and then looking for other type of splicing regulators, including, I'm not going to go detail, those exonic or intronic splicing enhancer or silencers, whether those variants are located in those regions, okay? And there is a paper published in 2005 in Science, and the tool called spinner, I think there is a spelling mistake here, but I will try to fix it. But the point here is that they use deep learning type of approach and in trying to predict whether one SNP occurred will cause the splicing outcome changes. So that is the tool that they are using. Okay. So now this is being connected to what we are doing. So what we are doing here, this is some of the study in my lab. So what we look at is from a slightly different angle. So we rely not only on whether this SNP is going to change the splicing, so sometimes may change, but once it changed, if the middle exon is being spliced differently, whether this middle exon encode an important part of the protein that will impact the structures of the protein or disorder of the protein and so on. So those are the structure information that we are going to use. So the way we did that is we look at, we took a very classical machine learning approach. We have a training data set. We have a bunch of disease-causing mutations. They are positive mutations. We know these synonymous SNPs, they are causing disease from a database called the Human Gene Mutation Database. And then we also take a bunch of negative ones, which are coming from the 1,000 genomes data. We know that they are not important. I mean, they are coming from the healthy individuals. So what we do is we evaluate these two types of SNPs. We want to see what distinguishes them, what makes them different. Okay, so there are many different things we look at. We look at whether this SNP is going to cause the splicing changes. There are a couple of dozen or hundred of features about that. And there is a bunch of a few dozen of features by looking at if the splicing is being changed, whether that middle exon is, what are the protein structure information on those. So after doing that, we will turn this into a tool, and then if we have a new list of the synonymous SNPs that we want to see what is the likelihood that they are associated to certain diseases. That's the way that we do it. And I have to show off some of our results. So this is the results, the ROC curve from that science paper that offered, and this is based on what our predictor that reported. And you can see that for both the variants on the splicing side and within the middle of the exon, so we all performed much, much better. I think the reason is it's not that we are doing better on the machine learning, and actually they are using deep learning, very fancy stuff. But this is really because we are using additional layer of information about the structure level information that the other study was not using. So I think that's why that we received a much better performance. And similarly, that we also developed another tool that looking for the SNPs in the intronic regions. So you can see that, I mean, the color is not very nice, but the outside ones is the one that offered by our predictor, comparing to two others using those existing tools. Okay. And of course, that those are the synonymous. Those are the SNPs that potentially will change the amino acid, change the splicing outcomes. So there are also SNPs that in the 3' UTR regions, and they can impact the microRNA binding sites, especially for the position, the first 8 positions in the microRNA. Those are seed sites. If they are being altered, and the chances microRNA targeting will through a very different mechanism. If they're in the promoter regions, and they can be considered as where the transcription factor binds. For example, there are 2 scenarios, and if this SNP causing this transcription factor binding, affinity dropped, and then that will influence the gene expression level for sure. For this type, it's a area that many tool has been developed on, and most of them just looking for the overlapping between the SNPs and the transcription factor binding sites. Either it's experimentally determined or bioinformatically predicted. Okay, so there are a bunch of tools that are being used here, and there are some other features being used. The limitation of all these tools is they are not really considering the degenerate features of transcription factor binding sites. So what I mean by degenerate feature is if this is the transcription factor binding sites, and actually there are many different letters here, and some letters are more important than others. So meaning that if this G is being altered, the chances this TF is not going to bind. But if this G is being altered, chances is fine. So different letters, they are having a different, I would say that information that in this TF binding. And we will have a very thorough review in this topic in the ChIP-seq session, because that is what the ChIP-seq after we identified the transcription factor binding, after the experimental consideration, we got to look at the transcription factor binding efficiencies. Okay, so I'm not going to go detail of this. This is another of our paper. So basically, in this paper, we're looking for the disruption of those binding matching score calculations, and this is the results. So for one of the studies, we found that those are the variants that being targeted we need to pay more attention to. 3 more minutes. Talk about the small insertion deletions, and those are have 2 types. One is the frame shift ones, so meaning that they are not a multiple of 3. And which means that if you change have a insertion deletion there, everything afterwards, the amino acid sequence will be randomized, and because the frame is being altered. And there's another part which is a multiple of 3, and those are 2 type of frame shifting indels. So this is one of our tool as well. So you can see that the top is deletions and the bottom is insertions. So we also look at in this scenario that this is red ones are the disease-causing indels, blue ones are the neutral indels, which means they are not causing the disease. But you can see that in terms of DNA conservation, we see very, very clearly disease-causing mutations are in the highly conserved regions. Those are important. Someone is texting me, so it's not mean that my time is up. and for this particular one, and then you can see this is ASA. So the so-called ASA is whether this is inside the protein or outside of protein. And we mentioned this topic point in the earlier slides. But what we want to see here is the disease-causing indels tends to be buried inside of the protein structures, while the neutral ones is outside. And this is a structure or disorder. So you can see very clear difference that disease-causing variants or indels tends to locate in the structured regions compared to the other ones. So after putting these together, and we're able to develop a very, very good predictor to identify what are the important small insertion deletions. This work has been published a couple of years ago. Okay, so far that we have worked on, we published a couple of papers on this area, and I'm not going to go too much detail. So the last piece I want to show here is I'm showing you a lot of different predictions for SIFT, polyphen, for all those tools that some other groups developed, we developed, and many of them actually is you don't have to download the original tool to do the prediction, and they can be, there is another wrapper called ANOVAR. So this is a very nice software. And you can basically, you have your VCF file, all those variants, and you can run very minimum type of command to get all these variants being annotated. And it's a very convenient tool. And also that in the ensemble database, there is a VEP online-based tool. So I'm not familiar with any of the Linux command or things like that, but this VEP will offer an online version, so meaning you can upload your VCF file, and they will help you to do the annotation. And also many popular tools have their own wrappers, like SIFT, polyphen, and CADD, and there is also other customized approaches. So last slide, this is it's being connecting with next week lecture. So after genome-wide association study or whole genome sequencing or whole exome sequencing, after the statistical genetics, and today we talk most focus on the bioinformatics predictions on the variant prioritization side. But regardless of how accurate you claim your bioinformatics tools are, and the chances nobody believe you that only based on the bioinformatics prediction. So in many cases, we still need to take this back to the labs and then to do a certain level of experimental evidence so that we can really convince people can spend more resource to manipulate individual variants and to see the phenotype. So in the next lecture, she is going to talk about high throughput report assay and the validation side by looking into how we can tackle this by screening thousands of variants in one single experiment. So that will be a very fun topic as well. Okay? And the next lecture, this coming Friday, and we will have a guest lecture from Dr. Matteo Vata, and talking about regulatory and ethical considerations in the clinical sequencing practice. Okay, all those different rules that research sequencing normally doesn't follow. Okay, all right, we are done for today. Have a good week, not weekend. Do you want to do it?
So I'm going to be posting a homework.
Oh yeah. Hold on a second. We have some. Ed is going to talk about homework. I forgot.
Yeah, so right now, actually, I'm going to put up the homework assignment. It's probably the only homework assignment you're going to have in the course, I think, for this
I don't
semester.
think so.
You don't think so?
There will be more.
Okay, there's going to be more. But this one, what I did is basically I set up a pipeline for you guys to run. I ran through a bunch of commands that are very simple. So you'll see the options on each step. And I wouldn't add any additional options, just take a look at what I've done. But while using these tools, look at the capabilities and what other options are available for them. And I made the data set smaller, so the jobs should run within a few hours, so that way you're not going to be waiting around for 2 days for an alignment to complete. So I've done what I can to streamline it for you. And all the tools in the assignment are available as modules, so you don't have to specifically install anything on your own. But that's going to go up right now. And the last thing I want to say is don't wait to start this assignment, because you're going to be competing with the entire school for compute time on these clusters. So you could run into a queue that's a week long because 3 or 4 PIs decide they want to rerun their whole data sets. So you need to know that your stuff is going to be working and that you're going to be able to get this done. And it's going to be due November 5th, which should give you a month before your final project is due. That's all.